Hello and welcome to the second short video in our series that I'm calling Factors that Impact the Activity of Enzymes. So this is part 2. The references that I'm giving you here are going to be exactly the same as the references that I gave you for the first part. So again, the Saladin text is very good. The WISE manual, not so good. OpenStax is also very good. All right, so let's get started. So we go now to the need to know. Specifically here in this video, what I want to talk about are the roles of different factors, including particle size, temperature, and pH on enzyme activity, and specifically on digestive enzyme activity. So these are things that relate to the enzymes and the substrate that make them work or not work so well. Let's talk about that. So we go back now to the image that I gave you in the previous video of an enzyme acting on a substrate. And so you can see again here, just to quickly reiterate, here's our enzyme and here's our substrate. And we have a very particular, a very specific interaction between the substrate and the enzyme in order to get the enzyme to catalyze a chemical reaction and work properly and make a product from the substrate. So basically, anything that enhances or optimizes the ability of the enzyme to interact with the substrate or bump into the substrate can be thought of increasing the activity of this enzyme, and thereby, if we increase the activity of the enzyme, we're going to increase the amount of product that's formed, or we're going to increase the rate of product formation. The specific factors that we're interested in this lab in looking at are going to be the effects of temperature, the effect of particle size, and the effect of pH on the ability of the enzyme to act on a substrate and to form a product. So in the case of particle size then, what we really mean is the size of the particles of the substrate. If we have smaller particles of substrate, then we have a greater surface area. If we have a greater surface area, then we've got more places for the enzyme to encounter that substrate. And if we have more ability for the enzyme to access or to encounter that substrate, then it's likely that we're also going to have more product that's going to be formed due to a greater enzyme activity. As a non-enzymatic example of this, I want you to think of the last time you had a Coke, and a fountain drink specifically, and you put ice in your cup first and then you put the Coke on top of the ice. If you had ice cubes and if the Coke was warm, you may find that it took longer for the Coca-Cola, your drink, to cool so that it was refreshing and delicious. If you had ice cubes, then if you had crushed ice in your cup, the crushed ice has got more surface area, so more ice can interact with the Coca-Cola. It can cool down the Coke more rapidly, and then you can enjoy it more quickly because it's cooled down more rapidly. The second factor that we're going to be talking about will be temperature. And what we mean here is the temperature where the reaction takes place. Now, most of the time when we're talking about chemical reactions, we usually think of them as going faster when the temperature increases because the substrate and the enzyme have higher kinetic energy, or they have more energy associated with movement when the temperature goes up. When they have more kinetic energy, they're more likely to bump into each other and then form a product. And it's everything about increasing the activity of the enzyme is about making the substrate and the enzyme more likely to interact with one another. Now, in the human body, we don't have too many places where we've got variation in temperature, and we certainly don't have any place where you have a large variation in temperature. But we are interested to see what changes could be produced in enzyme activity if we did have a big change, and that's really one of the purposes of our lab. Now, if our temperature for the chemical reaction is occurring where our enzyme and substrate are interacting is too high, then our enzyme can become denatured. And what I mean when I use the word denatured is I mean that the shape of the protein can actually change. The temperature can alter the bonds that are kind of holding this enzyme into this shape with these active sites. This is going to be that very particular place where this very specific interaction between the substrate and the enzyme is occurring. If we change the shape of the enzyme because we've changed the temperature, then this means that it's going to be less likely to have this optimal kind of interaction between the enzyme and the substrate, and that would decrease the enzyme's activity or decrease its ability to catalyze this reaction and form those products. The third factor that we're going to be talking about in our lab will be pH. And pH, or acidity, can alter the shape of the enzyme in much the same way that temperature can alter the shape of the enzyme. So when we change the pH of the surrounding medium, we actually can alter, again, those bonds that hold this protein, this enzyme, into this very particular configuration where our active sites are here just waiting for our substrate to interact. So by changing the pH, then, you might expect to see a decrease in the activity or some other change in the ability of the enzyme to produce a product. Okay, so the next thing I want to show you here, this is a table that I've created for you. Let me orient you to this table. So over here in the left-hand column, I've actually got the site where the enzymes are found, or different regions of the digestive system. So we go from the mouth to the stomach to the small intestine as you go from top to bottom, right? Almost like a trip through the gastrointestinal tract. And then here I've got three columns that are dedicated to different substrate digestion. We've got carbohydrate digestion, we've got protein digestion in this column, and we've got fat digestion here in this column. Okay, so what I'm showing you here are the enzymes that are present in each of these places. So we've got some amylase here, salivary amylase in the mouth. And I'm also showing you right underneath in black ink the reaction that's occurring. So in this particular case, you have salivary amylase. When we have starch in the presence of this enzyme, then we're going to produce maltose. Maltose happens to be a disaccharide, a smaller sugar, which is one of the breakdown products that we talked about in the very first video as well. So I want you to see that we've got, in some cases, we have the same enzyme or some similar enzymes that show up in multiple places. So we've got amylase here in the mouth, salivary amylase. We've got pancreatic amylase as well. And in some case, we've got enzymes that may have slightly different names. So we would call these amylases because they act on starches. We could lump these together and call these enzymes proteases if we wanted to, but they have specific names. So we call them pepsin and trypsin, and they happen to be located in different places. Likewise, we can have dipeptidases. This would be an enzyme that breaks down a dipeptide or two amino acid chain into single amino acids, and we also have aminopeptidases as well. So we've got similar enzymes that show up in different names. And the last thing I want to show you, in case you weren't familiar with this, is that you know that you're talking about an enzyme very frequently when the name ends in ASE. So we have lipase, dipeptidase, sucrase, lactase. That kind of clues you in that you're talking about an enzyme here. Okay, so again, we've got the enzyme, and then we've got the reactions that are being catalyzed here by each of these enzymes in each of these places. The next thing that I'm going to show you, this is actually kind of an overview of the digestive system here. It's kind of a cute little picture. And what I want you to see is that as you move through the digestive tract from the mouth, through the esophagus to the stomach, to the duodenum, to the small intestine, the large intestine. Over here, this author is showing you what the pH is. He's also giving you an idea of how much time foodstuffs spend in each of those places, which is not really what we're going to talk about, but it is kind of interesting. So I do want you to see right off the bat that in the mouth, saliva's got a pH of 6.5 to 7.5. As we go to the stomach, then we see that the pH drops dramatically, and the pH in the stomach can actually be below 1.5 if you've just had a meal. In the duodenum, then, which is this region of the small intestine that's adjacent to the stomach, it's just proximal to the stomach, what we see, or distal to the stomach, excuse me, what you see is that the pH actually rises pretty dramatically from 1 to 2 to 7 to 8.5. And then we see, again, it decreases again in the small intestine and large intestine, anywhere from 4 to 7. So we've got some very acidic regions of the gastrointestinal tract in the stomach, and then we've got some other regions that are actually quite basic, like in the duodenum. Now, a second ago, we said that pH will play a role in influencing the activity of enzymes, and so you'll want to keep sort of the ambient pH of these different regions of the digestive system in mind when you're thinking about digestive enzyme activity. I've also created for you another table that you can fill in. I've put a link to this, and I've put a hard copy on ELC. And if you look here, what you see is I've left blanks here for you to fill in the enzyme name. I've given you a hint: produced by the salivary glands, produced by the gastric glands, produced by the pancreas as an example. The site of action, where you expect these enzymes to act, and then the optimum pH. So the previous slide may help you kind of fill in this table as well. And I think that you'll find that a table like this will help you kind of keep track of some of the enzymes, or some of even the groups of enzymes that we have been talking about. So this brings me to the end. What should you do now? Watch the last video. That'll be part 3. Thanks very much.